Hello students, how are you? I hope you all are safe at home. Today I am here with Biomolecules part 1 of 11th standard. In this session we are going to discuss biomolecules of cell, classification of biomolecules, amino acids, types of amino acids, sugar, lipids, types of lipids, nucleotides, primary and secondary metabolites. So let us start with biomolecules of cell. As we know that each cell is composed of variety of molecules and molecular aggregates. The carbon compounds found in the cell of organisms are called biological molecules or biomolecules. For example, carbohydrate, fats, proteins, amino acids, lipids, etc. Now we will see classification of biomolecules. On the basis of molecular weight of biomolecules, it is classified into two major groups, that is micromolecules and macromolecules. When the molecular weight of biomolecules are less than 1000, then it is micromolecules. For example, amino acids, nucleotides, sugars, lipids, etc. And when the molecular weight is more than 1000, then it is macromolecule. For example, polysaccharides, proteins, nucleic acids, etc. Now it is about amino acids. Amino acids are the building blocks of proteins. These are organic compounds which contain an amino group and acidic group. So that is why it is amino acid. Why? Because it consists of two groups. One is amino group, another is acidic group. Amino acids are substituted methane with 4 substituent groups occupying the 4 valency position of the carbon. 4 substituent groups are hydrogen, carboxylic group, amino group, and a variable group symbolized by R. R group may be a carbon chain or ring. Altogether, there are 20 amino acids in protein. There is a specific characteristic property of amino acids. Amino acids are ionizable. Why it is ionizable? Because structure of amino acids changes in solution of different pH. So that is why they are ionizable in nature. Here it is about the structure of 3 different amino acids, that is glycine, alanine and serine. In all the 3, you may observe a variable group on the 4th side. Now let us see about the different types of amino acids. It is of 4 different types. They are basic, acidic, neutral and aromatic amino acids. Lysine and arginine are basic. Glutamic acid and aspartic acid are acidic. Alanine, glycine and valine are neutral. And phenylalanine, tyrosine and tryptophan are the example of aromatic amino acid. Now we will know about sugars. Sugar is the carbohydrate and simplest sugars are monosaccharides. Monosaccharides cannot be further hydrolyzed into smaller units. The monosaccharides usually have 3 to 7 carbon atoms per molecule. They have either free aldehyde or ketone group which reduce cupric ion to cuprous ion, called reducing sugar. Different examples of monosaccharides are glyceraldehydes, ribose, glucose, fructose, etc. The oligosaccharides are formed by joining together 2 to 6 monosaccharide molecules through condensation. And the bond formed between two monosaccharides unit is called a glycosidic bond. Here you can observe the structure of glucose and ribose. Glucose is hexose sugar, whereas ribose is pentose sugar. Now it is about lipids. It includes highly varied substances like cooking oil, butter, ghee, waxes, cholesterol, essential oils, natural rubber, some plant pigments, eucalyptus oil, etc. The lipids are not polymers, but they are assembled from smaller molecules by dehydration. They are non-polar and therefore insoluble in water. They disperse in water uniformly as minute droplets, forming an emulsion. Lipids dissolve in non-polar organic solvents like ether, chloroform, acetone, and benzene. They do not polymerize to form macromolecules, but they combine with carbohydrate and proteins. 3 to 5% lipid is present in the cell. Here you may observe the structure of fatty acid, glycerol, triglyceride, phospholipid and cholesterol. Now let us discuss about the classification of lipids. Lipids are broadly classified into three major groups. They are simple lipids, compound lipids and derived lipids. Simple lipids is further classified into neutral or true fats and waxes, whereas compound lipids are classified into phospholipids, glycolipids, lipoproteins and cutin and suberin. So we will discuss here one by one. First, it is about simple lipids. Simple lipids are esters of fatty acids with various alcohols. They are further of two types. First one is neutral or true fats, which are esters of fatty acids with glycerol. So they are also called as glycerides. Second simple lipid is waxes, which are esters of fatty acids and alcohol other than glycerol. Now it is about compound lipids. They are also called as conjugated lipids. Compound lipids are esters of fatty acids with alcohol but contains some other substances also. There are four types. First is phospholipids, which have a nitrogen containing base and a phosphate group in addition to fatty acids and glycerol. Second one is glycolipid, which have fatty acids and amino alcohol and a carbohydrate. Third one is lipoprotein, which have lipids, mainly phospholipids and proteins. Fourth type is cutin and suberin, which are cell wall associated glycerolipid polymer and they are specific to plant. Derived lipids are lipid-like substances such as esterol or derivative of lipids. Example, steroids, terpenes and prostaglandins. The steroids do not contain fatty acids but are included in the lipids because they have fat-like properties. Terpenes are a major component of essential oils produced by certain plants. Prostaglandins are hormones like compound derived from the 20 carbon polyunsaturated fatty acids. Their richest sources are secretion of seminal vesicles in male and menstrual flow in females. Now let us discuss about nucleotides. Nucleotides are the building block of the nucleic acids and it form about 2% of the cell contents. These are organic compounds with heterocyclic rings. Molecules of nucleotide consist of three smaller molecules of different types, that is a nitrogenous base, a pentose sugar and a phosphate group. A nucleoside has only nitrogenous base attached to pentose sugar. The nitrogenous bases are adenine, guanine, thymine, cytosine and uracil. Adenine, guanine, thymine and cytosine are present in DNA, whereas in RNA, thymine is replaced by uracil. Adenine and guanine are called as purines, whereas thymine, cytosine and uracil are called as pyrimidines. Polymerized nucleotides form DNA and RNA, which are the genetic material. Here you may observe structure of nitrogenous bases, nucleoside and nucleotide. Now the last topic of today's session is primary and secondary metabolites. Thousands of organic biomolecules are present in the cells, and this includes monosaccharides, disaccharides, oligosaccharides, fatty acids, glycerol, lipids, phospholipids, sterols, amino acids, nitrogenous bases, nucleotides, etc. These organic compounds have identifiable function and play a specific role in normal physiological processes and are called primary metabolites. Whereas secondary metabolites are product of certain metabolic pathways. For example, pigment, rubber, gum, resins, carotenoids, alkaloids, flavonoids, essential oils, antibiotics, scents, etc. These all are secondary metabolites. Many secondary metabolites are useful to human welfare and some have ecological importance also. Here there are some secondary metabolites. First is pigment. The metabolite is carotenoid, anthocyanin, alkaloids, that is morphine and codeine, terpenoids, that is monoterpenes and diterpenes, essential oils, that is lemongrass oil. In toxins, it is albumin and ricin. Lectins, it is concanavalin A. Drugs, that is vinblastine and curcumin. Now it is polymeric substances, that is rubber, gums and cellulose. So these are the some examples of secondary metabolites. So this is all about today's session. Thank you. For any kind of doubts, please write your questions in comment section.